Hello, we'll continue today the last two topics of clinical immunological disorders, the immunologic aspects of cancer and immunization. The immune system are more geared toward recognizing non-self rather than altered self. So tumors are self and lack PAMs, so there is no triggering of the immune response. Cancer cells escape normal controls that govern cell division and cell longevity. So, what are these rules? So, the transformed cell break the rules of governing multicellularity. So, cells normally don't divide unless given permission to do so by growth factors. Cells stay in their own neighborhood. A cell board in the skin stays in the skin, they don't move around. The cells live for as long as efficiently performing their intended function. Cells become injured, infected, or senescent, are killed or kill themselves via apoptosis and replaced. Cancers never arise from a single genetic mutation, but rather progress in a series of steps from normal untransformed state to a fully transformed one. The acquisition of a particular mutations will predispose toward acquiring additional mutation by generating and hence lead to DNA instability. Some of the key genes, such as B53, RAS, and c-Myc, once they are mutated and they are found mutated in a majority of cancers, these mutations can be accompanied by another additional mutation that are unique for each individual tumor. Oncogenes. So cancer arises from cells in which growth regulating and repair genes, which is called the proto-oncogenes, have become ineffective as a result of random mutation or following viral infection or chemical damage. When proto-oncogenes become altered or damaged, they are termed oncogenes, and their actions are capable of causing neoplastic growth. Examples of oncogenes and their action include first one is the P53, which is called the master regulator of the cell cycle. This encodes a nuclear phosphoprotein that inhibits cell division, thus suppressing tumor growth. So it's obvious that mutations in B53 result in uncontrolled growth. While RAS, this protein stays on the surface of the membrane and got activated by the receptor. It controls a GTP binding protein involved in signal transduction. Any mutation results in a failure of the GTPase inactivation of RAS. So once RAS is inactivated, it will have continuous activity, and the RAS have a lot of function that control the cell cycle. c-Myc, it's a transcription factor. The protein of the c-Myc gene, when translocated to a different, the gene c-Myc gene, when translocated to a different chromosome, it become oncogenic because this lead to the loss of regulation of B cell growth and the B cell lymphoma as well, Purkin's lymphoma. For BCL2 gene, this gene encodes a protein that inhibits apoptosis at normal concentrations. At high concentration in B cells, BCL2 promotes cell expansion and follicular lymphoma. The BCR-ABL gene fusion, two genes when fused to each other, result in a protein with increased tyrosine kinase activity, so increased phosphorylation for the other protein. This mechanism is involved in a chronic myeloid leukemia. Looking closely into B53, so B53, which is the master regulator of the cell cycle, normally exists as inhibited form when bound to another protein called MDM2. Once there is a DNA damage or any cell cycle abnormalities or hypoxia, this P53 is liberated from MDM2 and start to do two main functions. Start to do try to cell cycle arrest, and then the machinery of DNA repair works, and then restarting for the cell cycle. If this mechanism fail and there is no repair for the DNA, it will do apoptosis. P53 will promote apoptosis, so leading to elimination of damaged cells. In either two mechanisms, the P53 major role is keeping the stability of the cellular and genetic stability. While for RAS, normally RAS, it's a protein, G protein, which normally activated when bound to the receptor, the phosphorylated receptor that bound to the growth factor. Once the G protein is activated through a cascade of reaction involve a lot of protein kinases, a transcription factor will be recruited inside the nucleus that lead to the transcription of a lot of protein that stimulate a cell cycle, and this is a normal function of RAS. Once a mutation to RAS occur, this mutation may lead to that the RAS will be independently active and doesn't relate on the receptor for activation. This lead RAS protein will active with or without growth factor, so the same signal will be amplified, leading to the overexpression of protein involved in the cell cycle, leading to the cycling of the cells and the uncontrolled cell division. While for c-Myc, as we said before, it's any translocation of the c-Myc gene to the mu chain locus in Burkitt's lymphoma, as it happens in between the translocation between chromosome 8 and 14, the c-Myc gene, once translocated, is joined by this reciprocal translocation to the mu heavy chain. Hence, the normal mechanism of down regulation of c-Myc activity can no longer work because these genes moved from the normal chromosome to another chromosome where it cannot be regulated anymore. And so the cell will be held in a cycling mode, uncontrolled cell division. Looking into the normal function of c-Myc, it controls a lot of function like cell cycle progression, cell proliferation, differentiation site, genomic integrity, angiogenesis, metabolism, and apoptosis. Once there is a deregulation in the c-Myc, you will have a lot of abnormalities that favor cancer, like uncontrolled cell division, escape from immunity, cell immortalization, metastasis, and genomic instability and uncontrolled angiogenesis. Cancers are typically the outcome of cooperating mutations in oncogene and tumor suppressor genes. The key mutations that help to create the cancer state is called driver mutations, while other mutations may accumulate but do not generate a functional advantage for the tumor, and these are called passenger mutations. Driver mutations are typically gain of function mutations in oncogenes such as c-Myc and RAS, so that means overexpression of these proteins lead to a gain of function, or a loss of function mutation in tumor suppressor genes like B53. So the down regulation mutation will lead to cancer as well. The cancer development is a multi-step process. At each step in the progression, transformed cells acquire characteristics like the ability to grow independent of the growth factor, the resistance to apoptosis, and the ability to invade surrounding tissues. And more importantly, the adaptation that enable to escape from the immune surveillance, which is called immune evasion. These characteristics grant cells with a competitive advantage over neighboring cells and minimize detection or rejection by the immune system. Cancer cells arise from normal cells. In order for the immune system to attack a cancer cell, they need to be distinguished from normal cells. So we have different type of antigen associated with cancer. There are tumor-associated antigen, and they are not found exclusively on cancer cells. They generally present in higher amount in cancer patients and aid in diagnosis. Examples are the carcinoembryonic antigen and the beta-fetal protein. For the CEA, it's present on fetal gastrointestinal and liver cells. They are normally disappeared at birth, and it reappears in the serum of most patients with colorectal cancer. While for the beta-fetal protein, they are expressed in high level in patients with hepatomas and testicular keratocarcinomas. Their levels are lower in normal adults. For the second type of antigen is a tumor-specific antigen. They are unique to cancer cells and can be induced by viruses or chemical or physical carcinogens. They are either virus-induced TSA or carcinogen-induced TSA. For the first one, these virus-induced TSA, they are cross-reactive. That's say the genome of a particular virus synthesizes the same viral antigen in whatever cell they infect. Consequently, the immunotherapy should be applicable to all individuals infected by the same virus. While for the carcinogen-induced TSA, they induce random mutations in the genome of affected cells. Consequently, each mutated gene product, which actually is the antigen, differs depending on which gene has been affected by the carcinogen. So we have different antigen, different tumor antigen. So the immunologic cross-reactivity protection is not feasible. Immune response to cancer antigen. Immunocompromised hosts with diminished CD4 and CD8 cell functions have a higher incidence of lymphoproliferative cancers, implicating a T cell protective function. Macrophages are found frequently in the bed of regressing tumors. They must be activated by macrophage activating factor, example interferon gamma, in order to eradicate tumor cells via the respiratory path, nitric oxide, and tumor necrosis factor alpha. NK cells kill cancer cells through ADCC and lysis following contact. Their cytolytic activity is increased by interleukin 2, 12, and interferon gamma. Monoclonal antibodies directed to tumor-associated and tumor-specific antigen can be infused into a patient, either directly or conjugated with a toxin, a drug, or a radioisotope to kill the tumor. The example of that is the humanized anti-CD20 for B-cell lymphomas. Cell extrinsic mechanism of tumor suppression. CTL and NK responses are the most effective in tumor killing. The requirement for NK cell-mediated killing is cells failing to express the normal complement of MHC molecules, cells showing expression of non-classical MHC molecules. Requirements for CTL-mediated killing. Appearance of an MHC binding peptide that is recognized by a T cell receptor. To fulfill this requirement, the tumor would need to produce new epitopes, which for example called new epitopes. The new epitopes need to bind with the MHC2 plus the release of DAMs to activate dendritic cells to ingest the contents of dying tumor cells for successfully presentation. The cancer problem from an immune perspective. Cancer deploy multiple strategies to evade and repel immune attack. Firstly, they typically lack the molecular signatures. They lack the PAMs. Second, if strong non-self-antigen new antigens do arise during cellular transformation, they are weeded out through the immune-mediating killing of cells. That's the immunosurveillance. It is highly unlikely that an immune response directed toward a single new antigen will be sufficient to wipe out all the cells of the tumor. Third, tumors frequently deploy a battery of strategies to repel immune attacks through the secretion of factors like interleukin-10 and TGF-beta, or surface expression of molecules such as CTL4 or BD1 ligands that switch off T cell responses. Fourth, tumors tame the immune system and turning it to its own ends via the production of factors that re-educate the tumor-associated innate immune cells to an immunosuppressive or wound healing type mode. Cancers lack PAMs and contain few non-self-determinants. The exception to this rule applies to cancers that are initiated by viruses. A new antigen generated by a tumor is likely to be ignored by the immune system unless presented by a mature DC. Otherwise, tolerization to this antigen will occur. Tumors may create a microenvironment where tolerization of a tumor infiltrating lymphocytes occur through the failure of DCs to express co-stimulatory molecules. However, tumors that release endogenous DAMs may activate DCs and become subject to effective immune attack that may result in tumor rejection or immunoediting of specific subsets of the tumor. Conversely, tumors that fail to release DAMs may be simply regraded as a self and may fail to elicit a significant immune response and can of course escape the immune system. Also, tumors actively foster an immunosuppressive environment through the recruitment of multiple cell types into the tumor stroma that can actively manipulate the immune response. These include macrophage, Tregs, through the secretion of interleukin-10 and TGF-beta. Tumors express molecules that switch off T cell responses. Tumors often actively tolerize TCs in the vicinity through the secretion of interleukin-10 and VEGF, vascular endothelial growth factors, as well as factors that can suppress T cell activation, proliferation, and differentiation, such as TGF-beta. A key mode of T cell tolerization in tumor microenvironment is the surface display of BD1 and CTLA4 ligands that act as a switch-off for activated T cells. Another important means of frustrating T cell response is the manipulation of the metabolic environment within the tumor, leads to the depletion of important amino acid, the tryptophan, which is required for the T cell proliferation. In this figure, you can see the tumor. It can able to either secrete a tumor antigen and can secrete as well VEGF and interleukin-10. This figure shows how the tumors can actively tolerize against tumor antigen. This DC can, because the tumor cannot, most of the tumor cannot show PAMs or DAMs, this will lead to the lack of DC maturation, and so the lack of co-stimulation, so the T cell is tolerized. While the presence of VEG and interleukin-10 will inhibit the maturation of DC, leading to its inhibition, as well will lack of this lack of the co-stimulation of the T cell, leading to as well tolerance induction. The presence of one of the important molecule BDL1 together with TGF-beta will lead as well to direct inhibition of T cell. In this figure, we'll see how the T cell checkup points molecules CTL4 and BD1 are frequently engaged by tumor to suppress anti-tumor T cell response. You can see here that the tumor expressing BDL1, this BDL1 will block the activity of BD1 on the activated T cell. You can see that BDL1 binds to BD1. This BDL1 mediated antagonistic effect on the T cell activation will lead to anergic T cells. So by this mechanism, it will escape the immune system. Here we will see a video explaining, illustrative video explaining how BDL1 on the...
When it comes to treating diseases like cancer, modern medicine has an impressive arsenal. And one of its most versatile weapons are Y-shaped proteins called monoclonal antibodies. Our immune systems already produce their own antibodies. They come in billions of variations, each matching a specific target, such as a particular toxin, bacteria, or virus. When they bind to their target, they send a signal. This bacterium is now marked for destruction. These naturally produced antibodies are pretty effective, but in the 1970s, scientists figured out how to mass produce them. They can take any specifically targeted antibody and make huge numbers of identical clones, which means you can produce monoclonal antibodies for almost any disease you can think of. Which brings us on to cancer. Mutated cells that start growing out of control can be deadly. But the body's immune system isn't defenseless. Meet the CTL. It's a type of white blood cell that can detect infected or mutated cells. And when it finds one, it secretes toxic molecules that initiate a self-destruct sequence. But the CTLs do sometimes need to be reined in. If they get over-enthusiastic, the immune system can start causing collateral damage, attacking healthy cells. Fortunately, the CTLs can be turned off by activating a molecule called PD-1. The trouble with having an off switch is that cancer cells can exploit it. Many cancer cells protect themselves with a counter receptor called PD-L1. This is where monoclonal antibodies come in. Scientists can use one of two types. One that binds the PD-1 molecule, or another that goes for PD-L1. Thanks to the monoclonal antibodies, all of the CTLs are fully activated and can blast away the cancer. This is just one way that monoclonal antibodies can be used. They've already proven effective against a whole range of cancers, and yet more are still being developed. With their ability to be targeted at specific diseases, monoclonal antibodies are turning out to be a game changer, and one of our most effective weapons in the fight against cancer.
Tumor surface will inactivate the T cell, and as well the use of monoclonal antibody to reverse this process. The process of using a monoclonal antibody towards BDL1 actually got the scientists who managed to do that got the Nobel Prize last year. So we'll see the video now. And this slide show a summary for the two scientists who got the Nobel Prize in Physiology and Medicine or Medicine in 2018 about releasing the brakes against a cancer. So by blocking BDL1 or CLT4, so that's why liberating the immune system to attack tumor cells. Immune editing and antigen loss subvert the development of strong T-cell responses. The early stages of cancer are frequently blunted through immunoediting by TC and NK cells that recognize a strong tumor antigens, new antigens. This is because during the course of cellular transformation, which may take many years, the tumor is placed under selective pressure by host immune system. Genetic plasticity of transformed cells lead to the immune escape mutants that have lost or minimized the feature that the immune system is used to recognize tumors. So the ability of the transformed cells to manipulate their antigen, the surface antigen, will help these these tumor cells to escape from recognizing from the immune system. This is done by immune editing. In this way, the immune system may exert a Darwinism selective pressure for cancer-causing mutations that are largely immunologically silent and cannot be seen by the immune system or immune surveillance, a process that has been termed immunoediting. For example, it has been found that experimentally induced tumor in mice are less immunogenic if they have grown under the immune pressure, because as we said, the immune pressure provoke a Darwinism that lead to the survival of the strong tumor that will not be able to be recognized by immune system. So it's a kind of the immune system exert a Darwinism selective pressure against tumor. However, if the same tumor that have been grown in mice in under immune pressure, if the same tumor are initially grown in immune deficient null mice, followed by transplantation into naive wild type mice, 50% of this tumor fail to grow, as compared to 100% if the initially grown was in wild type mice, which don't have a compromised immune system. So we can see here that inflammation as well can enhance tumor initiation, promotion, and progression. And immune editing can lead to the selection of a weakly immunogenic tumors. So you can see here, it's initially the tumor contains a population of different tumor cells due to the different mutations and surface antigens. So due to the process of TC and NK mediated killing, it will attack some of these cells, leading to and due to the effect of tumor immune editing, this will lead to the escape of some of the tumor cells. Ultimately, the tumor cell that managed to do a lot of immune editing will escape from the immune system. So solid tumors are perceived by the immune system as wounds that need to be healed rather than attacked. Solid tumor always recruit tumor-associated macrophage and neutrophils through the secretion of cytokines and chemokines like CSF1 and interleukin 8. Such cells can provide the tumors with abundance of cytokines and growth factor that can promote angiogenesis, trigger tumor proliferation, suppress anti-tumor immune responses, and upregulate the anti-apoptotic protein so that they will not go through the apoptotic pathway. TAMs as well can suppress T cell response through providing of CTL4 and BDL ligand. So they switch off these two receptors on the surface of the T cell, so leading to its inactivation. An inflammatory environment can promote the progression to malignancy and metastasis through the production of ROS and RNS, reactive nitrogen species, that can provoke DNA damage and thus generate additional mutations. Prolonged inflammation can also predispose toward the development of a tumor, cell death in the tumor due to the deprivation of nutrients and blood supply, can also generate an inflammatory environment through the release of DAMPs from tumors. Infection can enhance tumor growth and survival through the utilizing of NF-kappa beta dependent upregulation of anti-apoptotic protein that make tumors resistant to stress. NF-kappa beta activation can also lead to the production of cytokines like interleukin 1 and 6, which can have autocrine growth promoting factors which aids in the development of the tumor. You can see here that tumors are bristling with a multitude of immune escape strategies. You can see it have ICAM1, which lead to decreased to conjugate formation binding. It have expressing class 1 MHC, which decrease antigen presentation to TC. It have another types of receptor leading to the loss of TC epitopes. It can secrete interleukin 10, which lead to the induction of Tregs, which have a suppressive function for the immune system. It have BDL1 and 2, L1 and 2, as we said. It leads to the inhibition of T cell activation. It can lead to the tryptophan depletion, an important amino acid need for the proliferation of T cell and the induction of reg. It have as well FASL receptor that kill FASL positive TC cells. Inflammation within the tumor, as we said, the tumor environment can be caused by multiple factors. You can see here a population of tumor cells, which can be in the inflammation can be enhanced by infection, pre-existing inflammatory conditions, oncogenic mutations, senescent cells, cell death associated DAMP release. These all produce a lot of inflammatory mediators that lead to the recruitments of macrophage, neutrophils, and myeloid-derived suppressor cells. The tumors uses the cytokines released by this cell to gain advantage to escape, to further proliferate and escape of the immune response. The chronic inflammation as well can promote malignant transformation or tumor progression. So you can see here that if some of cells have a persistent infection, this persistent infection will lead to the secretion of interleukin 1, 6, and 8. This leads to the recruitment of inflammatory cells like macrophage and the granulocytes. As we said, they will make use of the cytokines they release. The chronic inflammation that never resolve, opposite to a good inflammation. This leads to the transformation of the cell, leading to the generation of a lot of reactive oxygen species and the reactive nitrogen species. This DNA damage induced by these ROS or RNS will produce more inflammatory cells, and this is the never stop inflammation that may lead to irregular proliferation. And the last topic of the clinical immunological disorders is immunization. Most cost-effective weapon available against infectious diseases. There are four types of vaccines are currently in use or undergoing testing. Live attenuated vaccines permit replication of the organism in the host, increase antigenic stimulation, while killed vaccines contain organisms that have been inactivated by chemical or physical means. The multiple doses of these killed vaccines must be given, and adjuvant might be required for a protective response. This will lead to provoke the immune system. Recombinant vaccines, like hepatitis B vaccine, its formulation required identification of an epitope involved in the organism's pathogenicity. Synthesis of the vaccine foreign engine follows isolation and expression of the gene coding of the epitope in appropriate host cells. The fourth type is plasmid DNA vaccines, under development based on the isolation of microbial DNA containing the genes coding for an antigen involved in pathogenicity. Potential advantage of DNA vaccines include stability, low cost, ease of production, and long-last protection. You can see from this table, it shows the recommended immunization schedule for person aged from 0 to 6 years. So most children at that age need to have this schedule of immunization for trying to avoid a lot of diseases. Safety concerns of immunization. The live attenuated vaccines include insufficient attenuation, reversion to wild type, contamination by the live organism or toxins, and unsuspected immunodeficient patient may lead to problems if it's not efficient attenuation. Killed vaccines include contamination by live organisms or toxins. Autoimmune or allergic reaction or incomplete killing may lead to severe consequences. Recombinant vaccines to date are associated with a few safety concerns. Plasmid DNA vaccines, continuous stimulus may lead to tolerance or autoimmunity. Thank you, and this concludes our lectures about the clinical immunological disorders.
When it comes to treating diseases like cancer, modern medicine has an impressive arsenal, and one of its most versatile weapons are Y-shaped proteins called monoclonal antibodies. Our immune systems already produce their own antibodies. They come in billions of variations, each matching a specific target, such as a particular toxin, bacteria, or virus. When they bind to their target, they send a signal. This bacterium is now marked for destruction. These naturally produced antibodies are pretty effective, but in the 1970s, scientists figured out how to mass produce them. They can take any specifically targeted antibody and make huge numbers of identical clones, which means you can produce monoclonal antibodies for almost any disease you can think of. Which brings us on to cancer. Mutated cells that start growing out of control can be deadly, but the body's immune system isn't defenseless. Meet a CTL. It's a type of white blood cell that can detect infected or mutated cells. And when it finds one, it secretes toxic molecules that initiate a self-destruct sequence. But the CTLs do sometimes need to be reined in. If they get over-enthusiastic, the immune system can start causing collateral damage, attacking healthy cells. Fortunately, the CTLs can be turned off, by activating a molecule called PD-1. The trouble with having an off switch is that cancer cells can exploit it. Many cancer cells protect themselves with a counter receptor called PD-L1. This is where monoclonal antibodies come in. Scientists can use one of two types. One that binds the PD-1 molecule, or another that goes for PD-L1. Thanks to the monoclonal antibodies, all of the CTLs are fully activated and can blast away the cancer. This is just one way that monoclonal antibodies can be used. They've already proven effective against a whole range of cancers, and yet more are still being developed. With their ability to be targeted at specific diseases, monoclonal antibodies are turning out to be a game changer, and one of our most effective weapons in the fight against cancer.